Hi, everyone. So I'll be talking today about the future of diagnostic tools in AFM. And I think the reason that I was invited to speak to you today about diagnostic tools in acute febrile myelitis is because it's really important to make the diagnosis, to be able to properly treat with effective therapies such as drugs or vaccines. So before I actually talk about the diagnostic tools, let's review kind of this key question of AFM etiology. Because if you don't understand the etiology of AFM or why any given patient may have acute febrile myelitis, there's no way to be able to effectively manage or treat the patient's disease. One thing is that when we did metagenomic next generation sequencing to look comprehensively for infectious agents that may be identified in CSF, we didn't find anything. And the trend is also true for other viruses, such as poliovirus, that we didn't find poliovirus in CSF stool rectal samples. West Nile virus or other arboviruses were not detected. And in fact, part of the evidence basically grew to show that enterovirus D68 may be the culprit, given that in 2014 and 2016, it was the most common pathogen detected from non-sterile sites that were primarily respiratory. However, enterovirus D68 has very rarely, if at all, been found in spinal fluid. So the key question, which has been, is enterovirus D68 a cause of AFM? I think that the scientific consensus is that indeed it is a cause, but it may not necessarily be the cause of AFM. As you can see by the slide here, in 2018, for instance, there were documented AFM cases where some of them were caused by enterovirus, a different strain of enterovirus called enterovirus A71 versus enterovirus D68, which really brings me back to the importance of diagnostics for this disease. So let's review some of the diagnostic tools that we have in our armamentarium in microbiology. We can culture the organism, try to grow it in culture. We can run direct nucleic acid testing to look for DNA or RNA from the virus, and this is typically done by polymerase chain reaction or PCR. MALDI has been used to identify bacterial and fungal organisms, not really relevant here. Electron microscopy for viruses is rarely, if ever, used for diagnostics. There is a possibility of doing antigen testing, but we also have the availability of perhaps some new kinds of tests specifically for detecting enteroviruses associated with AFM. And then they may include, for instance, serology and host response biomarkers. Metagenomic sequencing is also a very attractive way of potentially looking at spinal fluid to see if there's another cause of AFM that's not enterovirus D68 associated, partly because, as I said before, enterovirus D68 has not really been, it's difficult to find in CSF. So if we focus specifically on infectious causes of AFM with regards to diagnostic tools, I'll be talking today about the areas highlighted or the specific tests highlighted in red, where we have, for instance, PCR, CRISPR-based detection for rapid nucleic acid testing, notably that this will primarily apply to respiratory secretions and stool because, again, we can't find enterovirus D68 in spinal fluid. We can try metagenomic sequencing to rule out other causes, but more specifically for trying to get at the question of whether or not any given patient is infected with enterovirus D68, there have been some recent new tests that I'll be talking about, which include CSF antibody testing as well as CSF host response analysis. So if we think about this year in 2020, there have been 9 confirmed cases. So if there will be an outbreak this year, we're sort of at the very beginning of an outbreak. But let's go back to 2018, where there were a total of 238 total confirmed cases in 48 states. If you look at here by, this is from Morbidity and Mortality Weekly Report, you can see here that the laboratory results from specimens collected from patients during the 2018 outbreak really show that very rarely can you find this positive cases or you can detect enteroviruses in spinal fluid. In fact, in all of 2018, there were only 2 of 74, 3% of spinal fluids that were tested were actually positive. On the other hand, if you look at respiratory secretions, 44% of the time, there was a putative enterovirus that was detected in respiratory secretions from patients who had AFM. Another limitation, however, beyond the fact that we cannot detect it commonly in spinal fluid, is that the common tests that we use for detecting enteroviruses cannot really discriminate rhinovirus from enteroviruses. So this was a study that was published in 2016 in Journal of Clinical Microbiology, where you can see that among patients who had AFM, when they found it, the patients that were eventually confirmed enterovirus D68 positive, you can see here that the target that actually lit up on the multiplex test that was used here, this is the film array test, really pointed more towards rhinoviruses. So we're unable to specifically identify enterovirus D68, at least based on the common tests that we do in the clinical lab. So I want to introduce kind of some new ideas in terms of how we can perhaps be able to better diagnose enterovirus D68 or enteroviruses in general. One is the use of perhaps a CRISPR-Cas12a based detection. This was a detection platform that we recently published, Nature Biotechnology, for SARS-CoV coronavirus 2 or SARS-CoV-2. The general idea behind CRISPR-based diagnostics is the CRISPR-Cas system is actually incredibly specific and very sensitive. And the idea is that you can construct guide RNAs targeting pretty much any type of pathogen and even targeting human tumor cells. And the idea is with these guide RNAs, they very rapidly hone in on any targets that may be present in your clinical sample, whether it's a blood sample, a stool sample, or a nasal swab sample. The Cas enzyme will then cut basically the particular sequence. But in addition to that, specifically the Cas12a enzyme can also cut a reporter gene, reporter sequence, which will allow you to detect fluorescence. So the general idea is this is a rapid way to detect any targeted type of pathogen, such as enterovirus. So we've applied this, for instance, for SARS coronavirus 2, and in short, we developed about a 30 to 40 minute procedure by which you can take either extracted viral RNA, and subsequently we've shown you can actually go directly from a nasopharyngeal swab sample and be able to get a lateral flow readout, sort of a urine pregnancy strip, akin to a urine pregnancy strip, get a lateral flow readout on whether or not someone was infected with SARS-CoV-2 within 30 to 40 minutes. It's our hope that we can use this to develop a home-based test for SARS-CoV-2. And similarly, we're currently working on a similar test for enterovirus D68. But the general idea here is that we've done now clinical validation. This is actually currently under evaluation at the FDA for EUA emergency use authorization approval. But the general idea is that we can be able to diagnose the disease in principle within 30 minutes. And hopefully we can do the same with enterovirus D68. I want to talk about kind of a second diagnostic technology. This is called metagenomic sequencing. The general idea behind metagenomic sequencing is imagine that you can just look at all of the DNA in a clinical sample. The idea is that in principle, in a single test, you can identify all potential pathogens or all potential organisms by looking at their sequence. And we've actually developed such a CLIA-validated assay. So this has now been validated in the clinical laboratory, and it's only run from spinal fluid. But to give you an example, this was just a recent case report of how this could be used to identify unusual pathogens. This was a 70-year-old male with mild dementia, presented with rapid decline, became akinetic and mute over 1 month, presented with essentially fever for about 3 weeks, had a negative workup, was treated for potential autoimmune disease, although did have a negative workup, notably had a history of non-Hodgkin's lymphoma and had been on chemotherapy last cycle 2 or 3 years ago, was also actively treated with rituximab, and was from a rural area but not very active. And I can tell you that all of the workup done for this patient was essentially negative, but when we did CSF metagenomic sequencing, it's basically an organism that you probably would not be able to appreciate, at least a priori. This was caused by a bunyavirus, but not only a bunyavirus, but we also saw that this was actually a bunyavirus called Potosi virus. And one of the aspects of Potosi virus, this this has never been described in human illness. It is an arbovirus. It's spread by the Aedes albopictus mosquitoes, and it was first isolated in mosquitoes from Potosi, Missouri. But this gives you an example of how the use of a comprehensive test, although it may not be as useful for diagnosing enteroviruses, because as I said before, we can rarely find enteroviruses in spinal fluid, might be useful for identifying other causes of acute flossamyelitis or neurological disease. I also want to end by talking about two new technologies. One is the idea of using antibody testing or serology. So this was a paper that was published by Ian Lipkin's laboratory and colleagues at Columbia, where we can see that the group was able to identify antibodies to enteroviruses specifically in spinal fluid. But in addition to that, they identified specific peptides that were enterovirus D68 specific, not only from spinal fluid, but potentially from serum, raising the possibility that we could develop or we could have an antibody test that can diagnose D68 directly from a blood draw. In addition to that, my colleagues, Dr. Michael Wilson, Dr. Joseph DeRisi, they developed what we call a pan-serological approach for essentially looking at antibodies to all viruses simultaneously using a phage display technique. I don't have time to describe it, but in short, what can be done with this technology is taking a spinal fluid, being able to identify essentially antibodies to all viruses. And when they looked at a subset of patients who had acute flossamyelitis, they saw, for instance, that essentially all of the, or nearly all of the patients actually had reactivity or antibodies specifically to enteroviruses. So this is a very powerful technology because it allows you to look at antibodies to all viruses simultaneously. But there's still one big challenge, and this is the big problem. One is, as I said before, direct detection is difficult because we usually can't detect this virus, enteroviruses in spinal fluid. We can't discriminate enteroviruses and rhinoviruses. It's difficult to routinely test for enteroviruses, even from, say, nasal swabs. But the second problem with antibodies are that antibodies may not be as useful in acute infection because, as you can see here, there is a window period before which you can actually have detectable antibody, either in spinal fluid or in blood. So the key question is, how do we address the ability, or how do we address the challenge of diagnosing AFM in early illness, in early stage of illness? And so one of our attempts here is to develop what we call a host-based RNA sequencing classifier. The idea behind the classifier is we could sequence spinal fluid, RNA that's extracted from spinal fluid from affected patients, and we can, by looking at the host response, we can determine whether or not someone was infected by, say, a viral meningitis or was infected with the enterovirus-associated acute flossamyelitis. So we developed here, this is a model which has pretty good performance, about 89% specificity and 100% sensitivity in identifying patients who have enterovirus-associated AFM. In particular, we've been trying to address this issue of, can we actually use this model to distinguish between patients who have D68 versus enterovirus A71 associated AFM? And this is really important because, as shown by work by my colleague, Dr. Misakar, that the outcomes are quite different. A child who has enterovirus A71 associated AFM very likely will return to his or her baseline, will recover significantly, if not fully, with time, whereas those who have or infected with enterovirus D68 usually have a much poorer prognosis, long-term prognosis. So the idea behind this is that we've been able to identify specifically differential expressed genes in spinal fluid that can distinguish between D68 and A71 infection. And in short, I'll be going very quickly through these slides, but in short, we've developed a classifier that this is still very preliminary, but has what appears to be about 80% accuracy in identifying patients who may have D68 versus A71 associated AFM. This is directly from spinal fluid without having to identify the virus in either spinal fluid or in respiratory secretions. So I want to end by just 4 conclusions. One is that diagnostic testing for AFM is challenging. And as a result, the diagnosis is really currently based on a clinical case definition. PCR testing alone is likely insufficient for the reasons that we have outlined here. Indirect testing methods are really needed, including perhaps antibody testing and host response testing. And finally, I think that timely diagnosis is critically important. And the reason is because more aggressive interventions may be indicated if the diagnosis of enterovirus D68 specifically can be made accurately and can be made earlier. So I'd like to turn now to a novel proposal from Dr. Matthew Vogt regarding antibody treatment for AFM. Thank you.